Bienvenidos al Paraninfo de la Universidad de Salamanca. Gracias por asistir a este acto de elección magistral y entrega de premios de la Cátedra Extraordinaria Terapias Avanzadas Novartis-USAL, Universidad de Salamanca, en cuya mesa presidencial me acompaña la directora de Relaciones Institucionales del Grupo Novartis España, doña Natividad Calvente Cestafe, a quien quiero agradecer mucho su respaldo a esta línea de investigación, de trabajo, de divulgación también, de la mejor investigación biomédica, y el director de la cátedra, el profesor del Departamento de Medicina, doctor Fermín Sánchez Guijo. Gracias también por impulsar las terapias avanzadas, la medicina de última generación en el Hospital Universitario y en la Universidad de Salamanca. Nos acompañan los premiados, a quienes felicitaremos, ya lo hacemos, pero tendremos la ocasión de homenajearles. El director médico del hospital, doctor De Quero, el director del IFSAL y del Departamento de Medicina, profesor Rogelio González Sarmiento, y el decano de la Facultad de Medicina, profesor Carretero, y la secretaria general, que lo es también profesora de la Facultad de Medicina. Pero sobre todo agradecemos la presencia del profesor Jacques Galipó, el ponente, el conferenciante de hoy, quien abrirá con su cátedra magistral esta sesión de actividades. Le doy la palabra, en primer lugar, de nuevo con el agradecimiento institucional, a la directora de Relaciones Institucionales del Grupo Novartis España.
Pues muy buenas tardes, rector magnífico, secretaria general, decano de la Facultad de Medicina, director del Departamento Médico del USAL, director médico del Hospital Clínico de Salamanca, director de la cátedra, profesor Galipó, y premiados y todos los asistentes, muy buenas tardes. Bueno, es un placer para el Grupo Novartis estar aquí, en este caso lo represento yo. Es un placer porque realmente nosotros desde el Grupo Novartis lo que queremos, nuestro objetivo es reinventar la medicina. Y reinventamos la medicina a través fundamentalmente de la investigación que realizamos aquí en España, fundamentalmente investigación clínica, que se hace a través de todos los hospitales, de la red de hospitales que tiene el Sistema Nacional de Salud, con un foco muy importante en el área de oncología, pero con una inversión también muy importante, siendo uno de los primeros inversores en cuanto a investigación biomédica en España. Esto no quita que para nosotros es fundamental toda la parte de investigación básica y la investigación translacional y esa colaboración público-privada que podemos ejercer a través de convenios, de acuerdos y de formas de trabajar como lo que representa esta cátedra de terapias avanzadas. ¿Qué voy a decir yo de las terapias avanzadas? Que es mi talón de Aquiles. Empecé hace muchísimos años en este área, conociendo al director de la cátedra, al profesor Fermín, y realmente supone las terapias avanzadas, y lo estábamos hablando en la comida con el profesor Galipó, supone es el presente, pero es una de las áreas terapéuticas y de las formas de tratar a pacientes que no tienen resolución más importantes que va a haber en el futuro. Son medicamentos poco convencionales, son medicamentos especiales por su forma de elaboración, por su forma de fabricación, por su especificidad en el ámbito de su administración y dentro de los pacientes. Realmente estamos hablando de esa medicina personalizada, esa medicina de precisión que siempre hablamos, pero este es el ejemplo más concreto. Realmente las terapias avanzadas, tanto en el ámbito de las terapias celulares como son los medicamentos CAR-T, que es la primera realidad, o una de las primeras, porque ha habido otro tipo de medicamentos, pero que no estaban tan especializados y tenían tanta precisión. Es una de las terapias más prometedoras que hay en el ámbito del futuro, pero también está esa parte tan importante que son las terapias génicas. Esas terapias génicas que, desde el Grupo Novartis tenemos a mucha honra tener el mayor número de medicamentos de terapias avanzadas. Tenemos dos medicamentos de terapia génica, uno en el área oftalmológica y otro en el área de las enfermedades raras, y en el área de hematología en las terapias celulares. Pues es un placer para Novartis tener esta cátedra de vinculación y de colaboración público-privada, donde se fomenta tanto la formación a los profesionales sanitarios como a la ciudadanía, como también la investigación académica, como bien estamos o vamos a premiar esta tarde a estos investigadores que realmente han hecho unos trabajos magníficos. Desde el Grupo Novartis queremos agradecer la colaboración a la Universidad de Salamanca, al profesor Fermín Sánchez Guijo, como director de la cátedra, por su bien hacer y su buen hacer durante este año de andadura de la cátedra, y esperamos tener muchos años de colaboración fructífera con todos ustedes. Muchísimas gracias.
Bien, muchas gracias a la directora de Relaciones Institucionales del Grupo Novartis, doña Natividad Calvente Gestafe. Ella lo ha expresado de forma perfecta. La colaboración privado-público y la proyección de la investigación es el presente y el futuro de las terapias y la investigación biomédica. Todo esto lo representa el titular de la cátedra y el equipo de profesionales que están aquí también acompañándole junto a los premiados. Le doy la palabra al profesor Sánchez Guijo para que presente al ponente.
Bueno, muy buenas tardes. Rector magnífico de la Universidad de Salamanca, directora de Relaciones Institucionales del Grupo Novartis España, autoridades académicas y sanitarias, estudiantes de nuestra universidad y de otras universidades, compañeros, amigos que nos acompañéis aquí, también la gente que nos va a ver a posteriori en la grabación que se está realizando en este momento. La segunda lección magistral de nuestra cátedra va a ser impartida esta tarde por el profesor Jacques Calipó. Él es actualmente catedrático de hematología y de oncología en la Universidad de Wisconsin, en Madison. Es también vicedecano de desarrollo terapéutico en la Facultad de Medicina y Salud Pública y dirige el programa de terapias avanzadas en la Universidad de Wisconsin, y también es el actual presidente electo de la Sociedad Internacional de Terapia Celular. Él estudió medicina en la Universidad de Montreal, él nació en Quebec, se licenció en 1988 y comenzó su carrera médica en la Universidad McGill y posteriormente ha estado en la Emory University en Atlanta, y de ahí ya se movió en 2016 a Wisconsin. Desde el punto de vista profesional, realmente es un líder internacional en el empleo de células mesenquimales. Nosotros venimos trabajando con estas células en el hospital y en la universidad desde hace ya más de 10-15 años. Él ha contribuido de una manera muy significativa a los primeros ensayos clínicos en humanos y en un montón de estudios preclínicos y translacionales en modelos animales. También ha trabajado en otras áreas importantes de desarrollo científico como las fusocinas, pero esto no es el tema de su charla de hoy. Él pertenece a los paneles de selección de los proyectos más relevantes a nivel de Estados Unidos y de la Comisión Europea. Tiene más de 10 patentes. Sobre el tema del que nos va a hablar hoy, tiene más de 200 publicaciones indexadas en revistas como Cell Stem Cell, Blood, Nature Medicine, donde obviamente pues es lógicamente también uno de los autores más citados. So turning now into English, I would like to really thank you, Jacques, for immediately accepting our invitation back in October to be here lecturing in our city. With all these uncertainties that we have in the past about the pandemic, we were not sure if you were able to make it. But for us, it's a real honor to have you here, to be able to show you our city, our university, and especially to share with you all the projects and the ideas that we have on developing the mesenchymal stromal cell therapy in the future here in Salamanca. Obviously, this is one of the greatest interests in medicine right now. So we're really looking forward to listening to your lecture. You have entitled it Past, Present, and Future of Mesenchymal Stromal Cell Therapy, an area where you are probably top one leader in the world. So congratulations, and looking forward to hear your presentation. Thank you.
So, Mr. Rector, faculty members, officials, members of the audience, thank you so much for the honor of inviting me to your beautiful, historic, and living university here in Salamanca. It's a real honor to be able to speak to you all today. And in particular, I'd like to thank my new best friend, Dr. Fermin Sanchez-Guijo, who coordinated my visit, and of course Sanofi for sponsoring my flying overseas, despite the pandemic, to come and lecture here today. It's a real delight. So, let's see here. This is a standard American disclosure. I don't have any conflicts of interest to disclose here having to do with the content of my lecture. So all of you in your bone marrow contain a large amount of stem cells that produce blood cells for you for the rest of your life. And for the scientists, those are CD34. But there's another type of stem cell in your bone marrow that are best described as mother cells. They're niche cells that feed and nurture these hematopoietic cells to live and grow. But these niche cells also have very powerful immune modulatory properties. Now, in your bone marrow, these niche cells live adjacent to either arterioles or venules, and they express certain cell surface receptors that are important to identify them, but also their functionality. And their functionality is characterized by the production of certain chemokines and growth factors. An important thing to remember is these stem cells in your bone marrow are vanishingly rare. If I take your bone marrow and try to just purify those, they're very few. They're 0.3% of total cellularity. But they have a really interesting functional feature, is these cells express the receptor for a kind of fertilizer or a morphogen called PDGF. So when we put these cells in a Petri dish and we add serum as a broth, these cells start proliferating, and you can grow a huge crop of cells that are your own over a few weeks. And these cells deploy all these desirable properties of regeneration that we're looking for. So, you have these young, happy, healthy medical students and PhD students. If I pay them $200, they sign a consent, I can take a little bit of bone marrow, a few tablespoons of bone marrow. It's an outpatient procedure, easy peasy, put a Band-Aid. I take this bone marrow out of the syringe, and I put it in a Petri dish. And when I grow their stem cells in broth, within 2 weeks, I could get 2 billion with a B, 1,000 million cells. Within 2 weeks, I could get 1 kilogram of cells after a month. Enough cells to be able to treat hundreds, if not thousands of adults. And these cells can grow. And because they deploy these niche mother-like properties, you can envisage taking these and giving them as a transfusion product to help treat diseases that fall in 2 broad categories. Regenerative medicine, which is to repair damaged tissues, and immune modulation for autoimmune diseases like Crohn's disease, for example, or multiple sclerosis as another. Now, in 1991, these cells were first identified with a clinical utility, and they were called mesenchymal stem cells, or MSC for short. And at first, they were, because what was known about them is they can turn into bone in a Petri dish. So they figured if somebody has a bone problem, I can take their stem cells, grow them up, turn into bone, fix the bone. And Arnie Kaplan, who is at Case Western University in Cleveland, he's the first one who discovered that. And the idea was to develop these cells for bone repair at first. And later on, we stopped calling them stem cells, and this is really for the experts in the audience here. We start calling them stromal cells, because when you grow them as a crop in the Petri dish, they lose some of their stem cell features, but they maintain a certain phenotype of expressing CD44, 73, MONO5, and they maintain mesenchymal plasticity in vitro. Now, what this graph shows you is even though I would take her stem cells and grow them in Petri dish, the input of stem cells is probably a couple thousand. And all of them start growing, but over time, a certain limited number of cells overcome the whole culture. That's called clonal impoverishment. And this matters because the stem cells that Professor Sanchez-Guijo grows in his lab, he harvests after 2 weeks or so. So they're nice and polyclonal and lots of participants, whereas the industrial cells that are where they manufacture 10,000 doses, there's going to be what's called, again, this impoverishment in representation, which may affect their potency. So as academics, we work with these early cells, which are, I think, is a good thing. Now, with the modern tools, we can take those stem cells and we can ask, what are the genes that are expressed that are of meaningful importance? And here what we did is we took mesenchymal cells from patients that were suffering from a disease called graft-versus-host or Crohn's disease, and uniformly, when we culture adapted their cells, all of them expressed all these morphogens listed on the left. The experts in the audience can look at this list and see is my favorite molecule there. But many of them are characterized by being immunomodulatory, angiogenic, and pro-regenerative. The beautiful thing about these cells is in the Petri dish, we can mimic injury by adding certain hormones that really turns them into the Incredible Hulk. And they start expressing an additional series of proteins and genes that are profoundly immunomodulatory. Because their job in life normally is if you break a bone, these cells normally in your body mobilize to the site of injury, help repair, and calm down the immune reactions so that healing can occur. So when we grow a large crop of them, we're replicating that physiology, but at a pharmaceutical level. So this cartoon depicts, as simplistically as one can think of, how mesenchymal cells work. So we can grow a crop of cells. This is the depiction of a single cell. And we can stimulate them with interferon gamma, which is that way of fooling the cell to think into there's injury. And they produce all these factors that are secreted that have an impact on immune cells that either make you sick or might be important in repairing your body. And amongst these monocytes, which is a flavor of white cell that goes in your bloodstream, are particular target of this, and they work by making another hormone called IL-10. So there's this cascade of events. So the first question we asked is, what is the true role of your own monocytes in your body as part of the response of this transfusion of mesenchymal cells? So to test this, we started off with a mouse model of colitis. Colitis is like Crohn's disease. So these mice, what we do is we give them soapy water to drink. Sounds terrible, but anyway, they drink the soapy water, and it gives them really bad diarrhea. Actually, the diarrhea is so bad, it's called colitis. And that mimics certain diseases in humans like Crohn's disease. So we have this animal model, and we ask, if we give mesenchymal cells, does it impact the disease model? So what I have in this little graphic on the lower part here is the mice. They're called black 6 mice. You give them soapy water for 6 days in a row. During the time they're getting soapy water on day 2 and day 4, we can give them an injection of mouse stem cells. And then we take away the soapy water because you have to stop it and just give them fresh water, and we can measure disease activity. The mice lose weight or they're sick. So to determine if macrophages are important in the host response to mesenchymal cells, we used a pharmacological approach to deplete all the endogenous macrophage monocytes in the mouse. And we use a drug, it's called liposomal clodronate. You inject it in the mouse, and this drug poisons all your monocytes and rids them of your body. So now we asked, if we deplete all the monocytes and we induce colitis and we give mesenchymal cells, what happens? And what we found was this. So this is the weight of about an average of 5 mice that we gave them mesenchymal cells, nice recovery. This is the weight of mice that we just gave them salt water. They lost a lot of weight. If we depleted the macrophages, they did even worse, despite receiving mesenchymal cells. So this is the first in vivo data that shows us that if you give mesenchymal cells, they interact with your own monocytes to lead to a regenerative response, even though these mice have normal T cells and B cells. So the interaction with T cells and B cells is not sufficient. So we're scientists. When you hit something, you keep on digging, and you keep on digging, and you keep on. So the question we ask is, what is it that these mesenchymal cells produce after you inject them that interacts with the monocytes? And again, this is data published by another group. This is directly looking in vivo in mice, in the bone marrow. You see nothing. All these blue dots are cells that are quiet. But if you injure the mouse, the mesenchymal cells in the marrow of the mouse, endogenous, turn green because they express a hormone called CCL2. It's a reporter system. And this leads to macrophages leaving the bone marrow, which is where they normally reside in your body, because they circulate also. So we asked the question then, is it CCL2 that's being made by the MSCs that interacts with CCR2? CCR2 is the receptor for CCL2. Cells are machines, so it's all switches and levers. And if they become IL-10 positive macrophages. So to test this, what we did is we took mouse macrophages and put them in a Petri dish. And we took a crop of mesenchymal cells and collected the media in which they bathe and put that in the macrophages and asked, do the macrophages become IL-10 positive? And the answer is yes, robustly so. So MSCs produce stuff that when you put in macrophages, the macrophages turn into those desirable. But if you add anti-CCL2 antibodies to block that, you lose the effect. So this is one piece of data that CCL2 in the conditioned media is polarizing the macrophages. This is the reciprocal experiment where we blocked CCR2 signaling, which is at the macrophage end, and again, you had the same effect. So this data tells us mesenchymal cells, hers, that I'm growing in a lab in a big crop, is producing lots and lots of proteins, but one of them in particular is the queen of proteins, CCL2, because if I deplete that, it loses the effect on macrophages. And to prove this in another way, again, so this is the weight of a mouse that's got soapy water. If I just give them salt water, they lose a lot of weight. But here, if the MSCs are derived from a mouse that's genetically incapable of making CCL2, so the MSCs are completely normal, they do absolutely everything except make CCL2, the mice lose weight as well, whereas normal MSCs rescue. So this is a third piece of data in vivo that the CCL2 from the MSCs is important. So we figured, aha, we have the answer. It's CCL2. Forget about stem cells. I'll just make some synthetic CCL2 protein. It's small. It's maybe 50 amino acids. And I could just give you injections of CCL2, and I'll fix you. So to test our hypothesis in the Petri dish, we went back to the macrophages. And if we add the supernatant from MSCs to these macrophages, they make IL-10, which is our sort of way testing that. But if we added recombinant CCL2, purified protein, no MSCs anymore, didn't work. The recombinant CCL2 on its own, on macrophages, did not polarize them. What gives? So then it hit on us. Maybe the CCL2 interacts with something else, and they form a pair, and that's what works. So to test that idea, we took the supernatant from mesenchymal cells that are CCL2 knockout, so everything is there except CCL2, and to that we added back CCL2, and it worked. So this was a first proof that CCL2 on its own is not the answer. It's necessary, but not sufficient. There's another secret molecule binding with it. So we're scientists, we gotta keep on digging. What's the other molecule? So we used a trick. So there you can use something called an antibody that binds to the CCL2, and if the CCL2 has anything else stuck on it, the antibody pulls it all down. And then we can analyze everything that got pulled down with it. And what we discovered is if we do a pull down to CCL2 in the conditioned media, we're pulling down CXCL12 also. So this is a piece of data that shows us that CCL2 binds to CXCL12, which is a different molecule, and they live together as a pair, and that gets pulled down. And we went on to show in the Petri dish, if you take pure CCL2, pure CXCL12, put them together, they act synergistically to lysis macrophages. So this cartoon summarizes everything I just told you in the past few minutes, that mesenchymal cells produce factors, of which 2, CCL2 and CXCL12, form a dimer, and that acts upon macrophages via CCR2. And at the receiving end, we then asked, what is it that macrophages make that's really important? We assumed it was IL-10. But to validate that, we gave MSCs to mice that had been induced colitis that were IL-10 knockout. That means the mouse genetically is unable to make IL-10. And what we did, when we did that, this is a disease activity score. The higher, the sicker the mouse. If the recipient mice were IL-10 knockout, they were much worse off than wild type. So now this confirms that MSCs interacts with macrophages, and those macrophages need to have IL-10 competency to deploy their effect. Now, in this experiment, what we did is we used a special genetically engineered mouse that if the cell makes IL-10, it glows green. So we caused colitis, we gave them MSCs, and after a period of 72 hours, we went back in the mouse and took out their gut. And let's see, yes, so we focused on their gut here. And we asked, are there any green cells that make IL-10? And what we found is if in a colitis mouse you give salt water, there's nothing. But if you gave MSCs, there was a huge influx of IL-10 competent macrophages to the gut. So the MSCs that you injected, those niche cells, they don't go to the bowel. They tell the macrophages that live in the marrow to go to the bowel. And here we repeated our cladronate experiment, that if we do the exact same experiment in these mice and we pre-treated them with cladronate, which depletes the macrophages, we lose all the IL-10 positive events in the bowel. So really this is telling us that mesenchymal cells interact with macrophages that go to the site of injury, and there they produce IL-10, and it makes you better. Now, if we give MSCs to a normal mouse, because the question is, where do those macrophages come from? Because macrophages, they circulate a bit in your blood, yeah, but most of them are in your marrow. And what we found was, in a normal mouse, about 29.7% of all the cells are monocyte macrophages. But if you give them MSCs, it drops down to 20%. So MSCs will mobilize marrow macrophages that will secondarily accumulate in liver, spleen, and lung. So one could come up with a generic mechanism of action. So this is how we can conciliate that MSC administration or cell therapy with MSCs can work for a wide array of ailments. It's not because it does something different for each ailment. It does the same thing, which is to mobilize those repair cells that live in your marrow, that go to the site of injury, start making IL-10, and fix you. So that's why MSCs are good for GVHD, they're good for Crohn's disease, they can be good for stroke, for any acute or chronic tissue disorder. And this cartoon basically summarizes that. MSCs form a heterodimer of CXCL12 and CCL2, interact with macrophages, and these macrophages make IL-10 and will shut down T cells. Now, I said CCL2 and CXCL12. In blue are all the chemokines. There are 50 chemokines. But here, I put in blue all the chemokines that are made by MSCs the way Dr. Furman does them. If you add interferon gamma, it actually induces these additional chemokines in red. And all the arrows I've put here are theoretically plausible heterodimers that could be formed in solution. So though we identified CCL2 and CXCL12, there could be many other heterodimers that are important. And macrophages express CCR2, but as well as CCR5 and CCR1. So you can imagine there could be a lot of biology there. So this is really the sum of the mouse data I'm going to show you. Now I'm going to pivot to clinical trials and clinical use. So now I've given you some new mechanistic data that explains how MSCs work. So then my question is, what are we doing with MSCs? So the first clinical trial where MSCs were given as a transfusion product was in 1995, 27 years ago, by Hitler Lazarus at Case Western Clinic in Cleveland. And it was actually in the setting of autologous bone marrow transplant. Since that time, there have been a very large amount of clinical trials. These are the years. In 2020, the inventory was 914 clinical trials worldwide of MSCs, including some studies done here in Salamanca. And as you see, the source of the MSCs could be either bone marrow, umbilical cord, or fat tissue. They're the three most prevalent. And the disorders treated range everything from acute to chronic tissue injury, as well as inflammation. And I know here in Salamanca, you're very much interested in graft versus host disease, as well as arthritis, which is joint, which is the second most prevalent. And again, this is the source of cells, and the cells could be yours, or they could be somebody else's. And somebody else's cells is really useful if you had, say, a stroke. Let me tell you why. Because to grow a crop of your own cells takes at least 2 weeks. But if you have a stroke, you need therapy within the first 7 days to impact outcome. So you don't have a choice but to use somebody else's cells. And the typical dose is 1 to 2 million cells per kilogram. That's the dose. By the way, right now in your body, you have about 1 to 2 million stromal cells per body weight kilogram. So when we give you a transfusion of this stuff, we're giving you in a bolus, in one squirt, as many cells as you have in your entire body. But these cells are also activated because I told you about all these chemokines, and they don't do that in your body at resting state. So the point I want to come up to here is there have been hundreds, if not thousands, of papers published using mouse models, like the colitis models I showed you, and I'm guilty of that cure them. Hundreds of papers, mice cured with mesenchymal cells. Yet, though there have been 914 clinical trials in humans, there's only been one approval yet, because there's demonstrated efficacy, and that's a product called Allogeneic that was developed here in Spain. So why only one? You'd expect there to be 100 based on the mouse data. So it comes back when, the definition of insanity is doing the same thing over and over again, expecting a different result, and that's what's been happening. So the failure analysis is where pharmacology meets reality. So for all of the audience here, if you remember only one thing is like stem cells are like sushi, fresh is best. Okay, let me get with me here. You would never eat sushi that was put in the freezer last night. You're going to eat it fresh. So they're PhD students here. When they inject stem cells in their mouse, and they spend months and years on their thesis doing this, they inject perfect cells. They come in on weekends to feed them. They read them books. They do everything that's required. They make sure the cells are perfect, fresh, and happy, and they inject them. Yet in clinical trials, what is done is they put the cells in the freezer. And then they thaw the cells and give the cells within 4 hours of thawing them. Is the fresh going to be the same as the thawed? Even somebody who's not a scientist is going to say they're not going to be the same. But you have to prove that. So the other thing I want to bring up is the route of administration. Of all those clinical trials, 43% of them give the cells intravenously. Makes sense, But there are other routes of delivery that can be contemplated. And like for graft versus host, you see most of them are given intravenously. But what is the data that giving the cells intravenously as opposed to giving them under the skin like an injection of insulin, would that work better? So if you give human stem cells IV in a mouse, they all get trapped in the lung, 100%. And within 72 hours, they're all gone. A little bit go to the liver. That's it. They don't engraft, they don't persist. And this is injecting human cells that have been engineered to glow. It's called luciferase. And if you inject them IV on day 0, you can see here the cells glow in the lung. By day 1, they're almost gone. By day 7, they're completely gone. So when you give mesenchymal cells intravenously, they will persist no longer than 3 days. 3 days, that's it. If you give them under the skin, they'll persist a week, in the abdomen 21 days, or intramuscularly 104 days. But if you take the same cells and you freeze them and you thaw them and you give them back afterwards, I'm going to stick to the route here. Here what we did is a mouse model of colitis. We compared giving the cells intravenously versus under the skin. And in our hands, the maximum tolerated dose of MSCs given IV had no impact on disease. Only the cells that were given IP or subdermally. So IP is as good as subQ, but not much better than IV. And how about thawed live cells? Again, if you remember one thing, stem cells are like sushi. So when you thaw stem cells, the way everybody does, they're doing a good job. Post-thaw, the cells are not dead. It's not dead garbage. They're not dead. They're alive, but they're really badly injured. If you give live cells to a mouse, live MSCs, you have a huge improvement in disease activity. But if you gave the exact same cells that were frozen and thawed, no good. Might as well have sung a song to them. And if you give dead cells, of course, it's at least the same thing. And it's been felt a lot of the clinical, virtually all the clinical trials using the 914 used thawed cells. You know which trial did not use thawed cells, used live cells? The one that led to the Allogeneic product approval. That was the one major difference. And here, basically, as long as the cells persist, the better they're going to be in regards to a clinical outcome. And this is just showing that giving fresh cells intraperitoneally, see the blue signal persists up to day 10, but if you give the same cells thawed, so it's again, thawed cells, they're not complete garbage, but they work much less well. And this is basically the same thing, but subcutaneously. So why is it that thawed cells are no good? Well, what happens is when we freeze cells, we have to put in certain molecules that allows them to be thawed afterwards without them exploding. And those molecules alter the cell surface membranes in such a way that the cells are more fragile. The second thing that happens, and this is for the scientist, thawing the cells unleashes a heat shock response. So you're basically have heat shocked cells that upregulate a whole bunch of injury molecules. They have attenuated immune suppression. And if you're giving allogeneic cells, they're susceptible to lysis. They get killed by the immune system of the recipient. And last, if not least, and again, this is for the students and postdocs in the audience, when you freeze a cell, the cytoplasm undergoes a vitrification. It turns to glass, frozen ice. And mesenchymal cells and many other cells depend upon the actin cytoskeleton for structure, but the actin cytoskeleton is also important for trafficking of signaling molecules in the cytoplasm. So vitrification depolymerizes the actin cytoskeleton. So a thawed cell is like a bag of broken bones. And the actin cytoskeleton needs to reconstitute itself, and it takes 16 hours. So all these things added together are all bad features of thawed cells. So when you give them IV or IPO, they just get cleared. There's no persistence, and there's no persistence, they can't make CCL2. If there's no CCL2, you can't mobilize the macrophages and nothing will happen. So fresh is best, but fit is fine. So culture rescue means you can take them out of the freezer, but if you put them back in the broth in which we grew them overnight, they recover all their features like Sleeping Beauty. They're fine. And this is just a data confirming that here, that fresh cells are good. They can persist up to 7 days. Thawed cells, you can't detect them. But if you culture rescue them overnight before giving that back, they're perfectly fine. So this cartoon now depicts all the data up to now. Mesenchymal cells can be eaten up, and that can lead to some IL-10 effect, but that's not the most important thing. The most important thing is they make chemokines, heterodimers that license macrophages to make IL-10, and the IL-10 competency is important for the regenerative effect. So what about using your own cells versus somebody else's cells? So to depict that, your own cells, a black mouse to black mouse, MSCs from black mouse to black recipient, and here the white mouse and the black mouse are genetically completely separate. It's like not even a brother, it's like somebody else. That's allogeneic. So syngeneic, autologous, your own, allogeneic. And what we found was using the colitis model, you induce colitis. If you give either autologous or allogeneic cells, they respond. They respond. But if you reinduce colitis and you retreat them, only the mice that got syngeneic or autologous cells respond a second time. Those that get allogeneic cells a second time don't respond because they became immunized, and the immune reject the donor cells. So if you're going to use allogeneic MSCs, it has to be in a one-and-done format, like acute stroke or heart attack, or something that you're just going to give over a period of 2 week cells, and after that, that's it, that's all. If you want to treat something like rheumatoid arthritis, and you anticipate you're going to be giving a dose a month for 2 years, you have to use your own cells, or else after the second or third dose, you're just going to reject the cells. So the roadmap to success, that one success that came out of the Spanish effort, started off with a paper by Dr. Herreros, and I believe, Furman, you were part of that activity as well. So there they took your own fat-derived stromal cells, and if you have Crohn's disease, you can get this terrible, terrible complication, which is called a fistula, that between your anus and the skin, these tracts appear, and it's really terrible. And there's no treatment. So the idea was, if you clean that out and then you inject your own cells, but here they didn't do this in patients with Crohn's disease. They did this in patients that did not have Crohn's disease, that had cryptogenic fistulas. So there they were using their own cells, they were fresh, and they injected them in this fistula, but they were not related to Crohn's disease. Big clinical trial, didn't work. CX401, Celrix. Come in, Tygenix, they buy Celrix. They say, you know what? You explicitly excluded Crohn's disease, which is an inflammatory disorder. If we do this in Crohn's disease, it should work. And then they made a second decision, which was a business decision. We're not going to use your own cells because it's too complicated. We're going to use her cells and treat 100 patients. So they used allogeneic cells derived from healthy donors. So allogeneic cells, fresh Crohn's disease, and they gave 120 million cells in a local injection. It was local. It worked. What they found was, if they looked at these patients up to a year later, those patients that got injections of salt water, about half of them, the fistula came back. But those that got the MSCs, the rate of relapse was much, much lower. And that was sufficient to get EMA approval. But again, these cells were fresh. So here's the approval. Exit Tygenics, enter Takeda. It's a bit like Darwinian little fish, medium fish, big fish eating, you know. Now, CX401 was the grandpa study. CXC01 was a Tygenic study, and now what they're doing is CX611, where they're not going to give frozen cells IV. I wish them luck, but I'm concerned that they're going back. You know the Einstein slide I made? There have been hundreds of trials, hundreds given frozen cells IV. Doesn't work. So, yeah, I don't know. So lesson learned in the mouse, it's best to give fit MSCs, either subdermally, auto or allo is fine. Thawed or killed cells, forget about it. IV delivery, it's a bias against you. I think subdermal works fine. And you can't give a repeat allo administration. So I'm basically, again, my advice to the outstanding program you're building here in cell therapy, huge investment, good investment, because they're great ideas. There's things to consider. Route of delivery, and you have the opportunity to do so subdermally versus IV. You're using fresh cells, which is 2 thumbs up, and you got to be clever about who you recruit. And this is the last mouse data I want to show you is there are tricks to make these stem cells more potent. So how we grow them in a Petri dish using hormones like interferon gamma. So this is the colitis, the mice lose weight. If you give them salt water, if you give them MSCs, they get better. But if you give them super MSCs, pre-treated interferon gamma, they're even better. Which informs a clinical trial we're doing in the US. It's the first in the world using interferon gamma MSCs. They're your own, they're fresh, and we're injecting them in the salivary gland of people that have bad dry mouth as a complication of radiation therapy. So thinking about the future, autologous allogeneic both have a route, and one has to think about allogeneic more in the one and done setting as opposed to chronic administration. Autologous is permissive for both, so that could inform clinical trials in the future. And I'd like to close on commending the Spanish ecosystem. Nationally, you're organized amongst the engineering facilities. I think that's a credit to Spain. It's a credit to Dr. Sanchez Guecho, who was leading this publication that described this. I think you're leaders internationally in what you can do because of your coordination and your national system. What you need is the resources to meet your goal, and I'm looking at the administrators or the managers, if they're here. Support them, good investment. The future for the investments you're doing here is point of care, vein to vein ATMPs. So place of care is here. So I think there's a future for cells manufactured in hospital systems that are delivered at hospital systems as opposed to in complement to industrial cells. And of course, enhanced MSCs is going to be the next generation. So I haven't touched a mouse in 20 years. That's why God created graduate students and postdocs. So I was, my first 12 years of my career at McGill University, and this was my team at McGill University in Montreal, Canada. I'm Canadian. Galipo is French because I'm French-Canadian, but I figured I wouldn't speak to you in French because then nobody would understand anything, except maybe Madame from Novartis. And then I moved to Emory in the US. This was the Emory. So you see, McGill, it's like, concrete walls, and a Canadian system like the Spanish system. Then I moved to Emory, the School of Medicine, marble everywhere, but that's okay. Emory's in Atlanta, this is the team there. Now we moved to University of Wisconsin, which is a public university, so way more casual. So the picture there is going to be more casual of enjoying, of course, Bibita. Is that how you say it? Okay, there you go. This is the team now at the UW. And again, for the scientists in the audience, I do want to remind everybody I'm the incoming president of the International Society of Cell Therapy. 2022, our meeting is in San Francisco in May, but especially for the young ones and everybody else, in 2023, our international meeting will be in Paris. Be there or be square. Thank you so much, everybody, for your attention.
Muchas gracias por esta magnífica conferencia al profesor Jacques Galipot. Ahora me referiré a ella y vamos a proceder a la entrega. El profesor Sánchez Dijo va a proceder a la entrega de un diploma al profesor. A continuación procederemos a la entrega de los premios a los trabajos de fin de grado y trabajos de fin de máster de la Cátedra Novartis-USAL, para lo que le pido a la directora de Relaciones Institucionales del Grupo Novartis que nos acompañe junto al resto de autoridades académicas en la entrega.
Bueno, antes de proceder a la entrega de los premios, queremos obviamente agradecer a todos los candidatos que han enviado sus trabajos, que es una convocatoria de ámbito nacional y obviamente todos ellos han sido de un altísimo nivel y como tales fueron calificados en sus respectivas universidades. Agradecemos muchísimo a los premiados que hayan asistido a recoger el premio de forma presencial en este acto, a sus familias les damos la enhorabuena que también les han acompañado y vamos a comenzar con el premio TFG 2021 de la Cátedra de Terapias Avanzadas Novartis-USAL. Es para el mejor trabajo fin de grado que haya versado sobre cualquier aspecto que esté relacionado con las terapias avanzadas y que haya sido defendido en cualquiera de las universidades españolas en el curso pasado. El premio ha sido para el trabajo Daños colaterales de la pandemia COVID-19 en la estrategia del programa de trasplante de progenitores hematopoyéticos y de programa de terapia CAR en el Complejo Asistencial Universitario de Salamanca. Entrega el premio el profesor José Carretero González, que es el decano de la Facultad de Medicina de nuestra universidad, y el premio es para don Guillermo García Martín. El accessit del premio TFG 2021 ha sido para el trabajo Implementación de las normas de correcta fabricación de una terapia avanzada en una institución hospitalaria. El premio va a ser entregado por el doctor Pedro Gómez de Quero Masía, que es el director médico del Hospital Universitario de Salamanca, y el premio este año ha recaído en don Pablo del Cerro Rodríguez por su trabajo que ha realizado en la Universidad Politécnica de Madrid. Pasamos ya a los premios de los trabajos fin de máster y el premio TFM 2021 de la Cátedra de Terapias Avanzadas Novartis-USAL es para el mejor trabajo, igualmente, pero fin de máster, que haya versado sobre cualquier aspecto relacionado con las terapias avanzadas, defendido en cualquier universidad española en el curso 2021. Y el premio es para el trabajo Células madre mesenquimales, MAPS doblemente negativas como portadoras del adenovirus oncolítico y COVID-5 para el tratamiento del cáncer. Entrega el premio el profesor Rogelio González Sarmiento, que es el director científico del Instituto de Investigación Biomédica de Salamanca y también el director del Departamento de Medicina de nuestra universidad, que es la sede de esta cátedra. El premio es para doña Patricia García Rodríguez por su trabajo en la Universidad Complutense de Madrid. Y finalmente, el acceso del premio TFM ha sido para el trabajo CAR Designs to Improve CAR T-Cell Function by using CD28 or 41BB costimulatory endodomains and IL-15 are alpha chimeras. Va a entregar el premio la profesora María José García Barrado, que es la secretaria general de nuestra universidad, de la Universidad de Salamanca, y el premio es para doña Inés Sánchez Moreno por el trabajo que ha realizado en la Universidad de Navarra.
Bien, pues enhorabuena a las premiadas y premiados por este reconocimiento que refleja un esfuerzo investigador, seguro que bien dirigido por investigadores e investigadoras en sus respectivas universidades, como el que desarrolla el titular, el director de la cátedra, el profesor Sánchez Guijo, con determinación, con acierto, con resultados los que vemos. Gracias a la colaboración con Novartis, muchas gracias de nuevo a la directora de Relaciones Institucionales del Grupo Novartis en España y la inspiración de investigadores como, en fin, el profesor Jacques Galipó, que nos ha ofrecido una conferencia magistral reveladora de las posibilidades de estas terapias avanzadas. Se ha dicho todo sobre la colaboración público-privado y la colaboración entre el hospital universitario, los grupos de investigación, la Facultad de Medicina, docencia, investigación, aplicaciones clínicas, pero tal vez el mejor reflejo del sentido de la universidad, de la academia, del espacio de saber que se proyecta en esta lección magistral y acto de entrega de premios es la puesta en valor de lo esencial, la naturaleza, porque estas terapias avanzadas tienen una magia muy especial y distinta a otras, salen del propio ser humano. No se trata de ningún producto artificioso, particularmente sintético, sino que toma lo mejor del ser humano. Hay algo de poético en las terapias tal y como han sido expuestas por nuestro conferenciante de hoy, son estudiadas. A mí me lo parece, busco una versión humanista en cada una de las aplicaciones del saber y de la ciencia. Y esa capacidad de extraer de nuestro propio ser lo mejor para curar, ¿no es acaso el propio arte de la medicina? Así que las terapias avanzadas expresan materialmente en sus utilidades y aplicaciones con la frescura tan recomendable que ha sido indicada en la conferencia, la versión más profunda de la capacidad sanatoria del propio ser humano, de las personas utilizando material humano para curar a otras personas de patologías tan propias de nuestras sociedades, las características de la longevidad, que es una buena noticia, pero requiere aplicaciones, cuidados más esforzados y, por qué no decirlo, también costosos, que han de ser efectivos para conseguir sus resultados. Un hospital universitario como el que tenemos la suerte de disfrutar en Salamanca, un instituto de investigación biomédica como el IFSAL, con una cátedra de terapias avanzadas como la que dirige el profesor Sánchez Guijo, una facultad de medicina que es emblemática en nuestro sistema universitario, investigadoras e investigadores como los hoy premiados y quienes les preceden en las líneas de investigación expresan también la mejor trayectoria universitaria. Así que muchísimas gracias por esta cátedra Novartis-USAL, muchas gracias por un año de resultados y qué buen futuro nos protege con estas terapias avanzadas. Muchas gracias.